Long before microbes were ever identified, people suspected that tiny organisms were the agents of disease. Some 2,000 years ago, the Roman writer Marcus Varro observed, quote, "In marshy places, little animals multiply, which the eye cannot see, but they enter the body through mouth and nose and may cause grave disease," end of quote. Varro, of course, was right, but it was not until 1,800 years later, in 1677, that a Dutch scientist, looking through the lens of a primitive microscope, was able to finally confirm the existence of microbes. Some 300 years later, medical science was under the mistaken notion that it had beaten back microbes. With the advent of antibiotics and powerful vaccines, there was hope that infectious diseases might be eradicated. But that hope proved illusory. Of late, we've witnessed diseases like AIDS emerge unnoticed and silently from out of nowhere and become the leading killer of American men between the ages of 25 and 44. Then, more recently, we watched helplessly in horror as the Ebola virus caused nasty, bloody, and quick deaths in Africa. Contemporaries of mine who went to medical school were taught that tuberculosis was a disease of the past. Never once in their medical residencies did they see even a single case of TB. But now, thanks in part to AIDS impairing people's immunity, TB is back, and with a vengeance, cutting a swath through densely populated urban centers. And in Central America, dengue fever, a tropical virus thought all but eradicated, has now reemerged in a more virulent and deadly form, and there is fear it may soon enter the United States. So as we near the 21st century, man and microbe continue to be a war that is developing new and deadly battlefields. One of those on the front lines is Dr. Robert Gallo. He has spent his research life working for the federal government's National Institutes of Health in Bethesda, Maryland. There he has sought to piece together the puzzle of human viruses. For instance, it was once widely thought that viruses had nothing to do with human cancer. But Dr. Gallo thought differently, and in the early 1980s, Gallo turned this medical dogma on its head by discovering a virus linked to human leukemia. Gallo's work with viruses caused him to become interested in AIDS and the possibility that the disease might be caused by an unknown virus. In 1984, Gallo and a French team at the Pasteur Institute in Paris independently announced that AIDS was caused by HIV. Gallo was subsequently embroiled in a bitter controversy over whether his team or the French team had made the discovery first. Patent royalties were at stake, and charges of theft and fraud were tossed in too. After years of investigation and innuendo, Dr. Gallo was ultimately vindicated. With the dispute now behind him, Gallo is leaving his post at NIH to head his own research center, the Institute of Human Virology in Baltimore. I sat down to talk with Dr. Gallo during the last days at his NIH office. So, Dr. Gallo, I'm interested if the route you took to medical research, whether it was straight or whether it was tortuous. In other words, as a child, were you interested in science and medicine?
Not really, not as a child. But compared to when you make comparisons when other people tell you their stories, I think it was a little straighter than most people. I would say by my last year in high school, and certainly in college, I knew pretty much what I wanted to do, that I wanted to be in biomedical research, and even had an idea that I'd want to study the biology of blood cells. I was that fixed by the later part of my high school period, and certainly by the beginning of college.
So in high school, did you have a teacher that turned you on to the subject, or?
To come to almost bottom line, there is no one single person or one single event that I can think of. When reflecting on this a few times before in the past, yes, it's true, in high school I had a very good chemistry teacher that helped in make my interest a little bit towards chemistry. It was certainly the best teacher I had ever had up to that time, and it certainly stimulated me. But there was also a pathologist, a physician at a local hospital, who was the first, let's say, cynic I ever met, and I had not seen anyone in my own family quite like that, and it left a very lasting impression. And he personally, you could say, took me under his wing, and I used to spend time in the hospital with him, even in high school and certainly in college. This followed the death of my sister, who had leukemia, and he was the doctor who made the diagnosis. So he influenced me a lot. His name is Dr. Marcus Cox, and still in Woodbury, Connecticut today, still reasonably active.
When did you first look at a cell?
With him, probably in high school, yes, with his microscope, undoubtedly. Tissue from surgery, removal, tissue that had been removed, post-mortem samples. So no doubt in his laboratory and office in Waterbury, Connecticut.
So I'm curious, you did an article for us a few years ago at Discover, and you talked about some of the generalizations about what we thought about infectious disease a decade and a half ago. Yes. And then we reflected on how those generalizations had changed. Why don't we talk about that? I mean,
sure.
what did the world, I mean the world of medicine, think about the possibility of another epidemic in the so-called developed world, you know, looking, let's say, 1978, 1979?
Yes, it is an interesting point. I think there was overconfidence. As you know, I've written in that way. There was a general feeling that infectious disease was almost passé. It was something for the third world.
And the last epidemic the world had was during World War I.
Well, I think the real great epidemic the world had was, of course, influenza, 1918, 1919, the period of World War I or the aftermath. I don't know the numbers, but it's a big number. And those serious epidemics of influenza, of course, are not over. Depends on the strain and some other factors that emerge. But nonetheless, somehow or other, there's so much excitement in science in the 1970s, and there still is, in biomedical science, and so much in molecular biology, that it was a great temptation to feel, let's put aside infectious disease, at least in the United States. And I think to a lesser but still significant degree also in Europe, infectious disease for the third world. But when you analyze it, that attitude was a bit arrogant, even if it was only the third world, because that's part of our world. But then the microbes reminded us that with the changes in society that have occurred in our time, that there is no third microbial world. It becomes one microbial world, with some rare exceptions. Because of travel, certainly post-World War II, the enormous increase in airplane travel. The economy improved, the development of airplanes for tourists, the massive tourism. I think that's number one.
Overpopulation, too, must play into this.
Population, I think, plays a factor, and changes in Africa, for example, with the loss of the moving out of the colonial powers and people moving into cities because of changes of way of life, a massive increase in size of cities in Africa, I think played a role. The use of blood products going from not just one city to another city, but from nation to nation.
And I guess also at the end of the 1970s, people thought that if there was to be a worldwide infectious disease, that would have to be transmitted very casually, that if it were by sexual transmission or contact with blood products, that it would just be too slow.
Pre-AIDS, the general thinking would be that something to spread globally would be casually transmitted. It was forgetting or not realizing that some microbes could be around a long time, could be persistent and infectious persistently over a period of not days but years. If you're used to thinking about influenza, or if you're used to thinking about an adenovirus infection or a virus which causes the cold, let's say, as we usually think of viruses, it's something you quickly catch, you incubate and quickly get rid of, and you infect other people only in a very short period of time. But now you have a situation, let's imagine, where the infectious agent is with you for 10 years, as it is in AIDS.
So also at the end of the 1970s, sort of the received view was that there were no viruses that caused cancer in humans?
Yes, certainly it was the dominant belief throughout this century that viruses played no role in any human cancer. But by the mid-1950s, it had become evident that viruses in some animals could indeed cause cancer. And most of this was in laboratory setting. But by the 1960s, it became clear that viruses could cause cancer in nature in animals, in at least a few instances. cat leukemia virus being a beautiful
Right.
example discovered by Dr. Jarrett in Glasgow, Scotland. But then a good 20 years to 25 years, let's say a quarter of a century of a search, came up with virtually nothing. But by the end of the 1970s, it was almost taboo, or late 70s, to be looking for viruses that cause cancer. Then all of a sudden, by the end of the 70s, early 1980s, we had multiple examples.
Tell me about those. How did we find them?
Well, first of all, an old virus, known and discovered earlier than the viruses that we found, that I'll come to in a second. The Epstein-Barr virus, the known cause of infectious mononucleosis, EBV. It's a herpes virus. Herpes virus, never known to cause cancer. But by 1980, it became clear that EBV, this particular herpes virus, could cause an unusual form of cancer, mostly of children, Burkitt lymphoma. And it caused it in a complicated way because the virus is everywhere. But in combination with certain other things like malaria, it is believed, it is thought, certain genetic changes occur in the cell, which leads to this tumor epidemic in parts of Africa. So these things became clear that Epstein-Barr virus was playing a role, but it was complicated because the virus was everywhere, but the cancer was only here and in this population, so other factors were involved. And now we accept it as playing a role in the multi-step process leading to a cancer. But those were old viruses, not suspected of causing cancer, that in the early 80s, it becomes apparent that they're involved, again, as a multi-step process, again, probably involving something else. And exactly at that time, my colleagues and I discovered the first retroviruses, which we called HTLV1, then a second HTLV2, viruses that had long been sought for, because in animals, they were so often in multiple different species shown to cause leukemia or lymphoma. So they were sometimes called leukemia viruses or lymphoma viruses. And occasionally they caused sarcomas.
In a nutshell, what's a retrovirus?
It's a category of a virus. You know, first of all, what is a virus? And virus is nothing more than some set of genetic information covered by some protective surroundings of protein coats that has some usually specific way it interacts with the cell membrane. A retrovirus has as its genetic information RNA instead of DNA. That's not unique. That's true of polio, true of influenza. But now we come to what distinguishes the retrovirus is it also carries a special enzyme which allows the RNA to be what we call transcribed. For the purposes of this discussion, let us say converted into DNA. So when a retrovirus infects a cell, its RNA genetic information ends up in the form of DNA. That DNA gets to the nucleus of the cell and integrates into our chromosomal DNA or the animal's.
I see. So the virus almost harnesses the cells in our own body.
Right. Once you're infected forever, because when those cells divide, the daughter cells will contain the viral genetic sequences, what is called a provirus. And it can be there in a silent form so the immune system can't even see it. To go back a minute and finish the question of this period of no belief that viruses could cause human cancer, we start finding retroviruses for the first time. And people had to say, well, wait a minute. Now, these are viruses that cause cancer in animals. The very finding of them was almost evidence that it was the cause of some cancer where you found them, because they had caused leukemia in so many species. And very soon, our laboratory, in collaboration with epidemiologists and in work independently in Japan, established HTLV1 as the cause of adult T-cell leukemia. If you put all this together, you can say evidence is right now that viruses play a role in a very significant percentage of human cancer if you look at the world at large. Sometimes in a more direct way, sometimes only as one of many needed steps in the process to cause malignancy. What percent of the world's cancer involves a virus? I can't give you a precise number, but I would certainly say it's over 20%. It may be as high as 30 to 40%.
When did you first learn about AIDS? When did you first hear about people coming down with it?
1981 from the newspapers. I remember reading about it only as some mysterious phenomena in a small number of gay men. I guess I read about it from New York or from the West Coast. I'm not sure. I didn't remember details.
And then how did you get involved in doing research on the cause of AIDS?
Well, it's easy to recall that. It was stimulated by listening to a CDC epidemiologist, I think had played a major role in catalyzing America's awareness of AIDS at that time, Jim Curran from CDC. I heard him lecture a couple of times. And one time he was provocative, and some people tend to be easily provoked. And he said, where are the virologists in this problem? And this was before there was any knowledge that it was due to a virus. When I heard Curran, remember our lab was already working in T-cell biology.
Right.
We had found an interleukin, I guess the earliest interleukins, interleukin-2 T-cell growth factor, so we were growing T-cells in our lab regularly. And clinicians had argued that this was a disease of the CD4 T-cell, a special kind of T-cell. So we had a certain expertise in studying and growing those cells. Not more expertise than immunologists, but perhaps more expertise in growing them. We had.
And the T-cells being, just for people who don't know?
T-cell is a kind of a lymphocyte, like the B-cell is a lymphocyte that makes antibodies. The T-cell is another lymphocyte that's critical for what is called the cellular immune response system. And clinicians noted that in people with AIDS, the special kind of T-cell called CD4, or what is mostly referred to as a helper T-cell then, which does all kinds of things important for the immune system, and especially in fighting viruses, was declining. Okay, so presumably that cell was central to the disease.
And that's referred to now when people talk about T-cell counts.
Yes.
Okay.
And if that cell was central to disease, it was a cell I was studying and growing all the time, and probably was growing as longer than anybody in the world. And also, we were dealing, as you know, with virology, retroviruses, a little bit herpes viruses. But at the very time that we were becoming aware of AIDS, the leukemia viruses I've talked to you about already, HTLV1, we had just discovered another, HTLV2, and we noted that when they didn't make T cells grow forever in the lab, they impaired their immune function. We had evidence to believe they came from Africa. We had evidence to believe that they were transmitted by sex, by blood, and by mother to child. Therefore, I thought, speculated in 1982, that the best candidate as the cause of AIDS in my mind is a new retrovirus, probably related to HTLV1 and 2, maybe arising by a recombination of this virus with some animal virus in equatorial Africa. Now, the interesting thing is the general idea was the most fruitful and productive and was the closest to being accurate, but Mother Nature pulled a trick. The cause of AIDS is a retrovirus. It does target T cells. It almost certainly does come from Africa, but it has nothing to do with HTLV1 or HTLV2. A whole new family. So we were right and we were wrong. In any case, we started to work a little bit on AIDS because we thought the problem was not very important. Mm-hmm. There's often been a sentiment, probably with some justification, in the gay community that it was avoided to be studied because they were not in mainstream society. I can't say that's true or false. I can only tell you in this area, in this campus, in this laboratory, those things were never discussed. It was that it wasn't important yet because there were so few numbers we were hearing about. And this is the Cancer Institute. We had other things to study. And the thing that's interesting is nobody ever asked us to get involved in studying this disease, except that provocative statement by Jim Curran. In fact, the government, NIH, or anywhere doesn't have, and there's nobody that's, a new epidemic, you're responsible for it.
Right.
To this day. CDC is responsible, but mostly for tracking the epidemic and public health measures. But there is not a group to say you're responsible to find the cause, prove the cause, and develop
Should there be?
faster. Well, it's an interesting question. I think, yes, I believe that there should at least be some kind of organized effort on day one to say that there's a responsibility, particularly in health center.
There was the old war on cancer, I guess, in the Nixon years.
But this is a specific disease and a specific new epidemic.
Right.
So you could argue that the best thing is let chance do it. It happened, and there are people who argued that the fastest pace in medical science was 83 to 85, from the inception of a new disease to its understanding the cause, to the development of a blood test, to having the reagents, the chemistry of the virus pretty well known, its target cells known, the epidemiology known, and there's probably a lot of truth in that. But I regard it as also a lot of chance. We were working with one finger on the problem, And certainly nobody told the group in France to work on it either, from what I understand. It's all in fact, I know the story pretty well by now, and I know how they got stimulated to work on it. And part of me getting more involved was a French clinical immunologist, Jacques Leibovitch, who visited me saying he had read my proposal that this was likely to be due to a retrovirus targeting T cells. And he read about HTLV1, and he said the whole thing excited him, and he came to me with a canister of a half dozen or a dozen more samples from AIDS patients, and really was forceful in pushing us to, say we should be working more fully on the problem, telling me it was growing now in Europe, etc. And so we worked hard on the samples he brought to us, in fact. And he brought back some of the information from our lab across the sea. And a man named Paul Prunet of Pasteur Production or Pasteur Diagnostics, I'm not sure, took some of this information and said these ideas ought to be pursued, and stimulated people there in beginning of 1983. We'd already been working for about 6 months, but with, as I said, with one finger in 82. In retrospect, I think that with a little more care, we could have even been faster and had this thing done pretty well in 82, early 83. I would say we lost a year from some confusion. But in any case...
Confusion over?
Oh, confusion with ideas. With one of the samples, ironic, it's not often told, but again, it's enmeshed in my book, but Jacques Leibovitch, when one of those samples he brought us was a young Frenchman who vacationed in Haiti. His initials are CC in the story in my book. He's described in some detail in a recent book by a woman author as one of the first cases, if not the first case in Europe. The woman author, I think it's Suzanne Garman, doesn't know some of the real interesting thing about that case. He was brought to us in 1982, and he turned out to have been blood transfused after a car accident. And now he was one of the first in retrospect. One doesn't get credit for what one didn't perceive. But in retrospect, he was one of the first, if not the first, that we could grow the cells and show that had HIV. And what we were doing is growing two viruses unwittingly. He was double infected with the leukemia virus HTLV1, which is endemic in Haiti, and HIV. So we had a mixture of the two, which caused us enormous confusion, because we never considered that possibility. We considered that he must be a recombinant of the theory was exactly right, HTLV1 with some animal virus. And the cells were producing something that was reactive with our test material for HTLV1, yet the substance it produced could kill T cells, so like an AIDS virus. So we kept looking for that which was partly related to HTLV1 and still could kill. And then we found that only 8% of times, those are 8% of people who are double infected, usually drug addicts. And so you see but that confusion certainly set us back scientifically in that pathway for some time.
So was it in 84 when your team and the team at Pasteur pinned down the HIV virus as being a cause of AIDS?
By early 1984, the cause of AIDS was defined by us, yes. You could certainly say that, and the blood test developed. The Pasteur group reported in 1983 in a case report of a young man with lymph gland enlargement. And so they call the virus lymph node virus or lymphadenopathy virus. They didn't show it was linked to AIDS in this report, nor they didn't have data to say so, nor did they claim so. And they were cautious, and they couldn't say more. It was one case. And it was not a person with AIDS. The person had the lymph gland enlargement but had not evolved to AIDS yet. That virus is the right virus, but by the time that was proven and that we had many isolates of the virus and the blood test developed, it was indeed early 1984.
What we are publishing in a few days' time is approximately 90% of people with AIDS have detectable antibodies in their serum that react specifically with this particular virus.
Then was the cause was known, the virus was available in large amounts, probes of the virus were available, what we call reagents were available. Many people could come into the problem and work on it. But even then, some people were still working in other causes. And indeed, 1 month before Secretary Heckler made the announcement that we had the blood test and the cause in hand, my very institute, another institute at NIH rather, made an announcement that a fungus was the cause of AIDS, which is, it was interesting, and it's interesting how that happened. I mean, in a way, how you can be misled.
Yeah, tell me about that.
My assumption, from what I've heard, is that they had an accidental contamination with a fungus in several cultures. But that's not enough. What was exciting to them was that the fungus was making a compound that kills CD4 T cells, cyclosporin.
Right.
And I can remember vividly, I was in a hotel room in San Francisco when the director of the Cancer Institute, Dr. DeVita, called me, and he said, "I thought you told me it was going to be a retrovirus." And I can remember getting palpitations. And I said, "Yeah, our data is really strong now, and I'm convinced, and other people are getting data at the same time now, and it's coming together." And he said, "Well, you're going to see on television tonight that so-and-so is going to say that's due to a fungus." And he told me that it made cyclosporin. And I thought two things at once that settled me, and the palpitations stopped. One, I remembered Einstein's statement that Mother Nature may not be cruel, but she's never that simple. And the one agent we know to knock off T cells, cyclosporin, is going to be handed to us by a fungus. But more importantly, I also remember that the fungus won't pass through the filters. And factor VIII was filtered in a way that would exclude fungus.
A filter, you mean when blood is filtered?
Hemophiliacs receives a substance called factor VIII. In the preparation at that time, the material is filtered through a filter that would exclude a fungus. A virus could pass through, but a fungus could not.
Right.
Okay?
So you knew immediately.
I knew it had to be a fungus, right.
So obviously, recently there's been a lot of controversy over when you discovered the virus, whether Pasteur discovered it when, and, you know.
It's not recent.
Well,
ancient.
I mean, long after the actual discoveries were made.
Yeah.
And I know one of the things that most offends you about this is the people saying that somehow the competition back then, slowed AIDS research.
Yeah, I mean, it's almost absurd. I mean, of course, nothing is perfect. But to talk about slowed, competition almost always accelerates research. If you're talking about instant communication, the day you do an experiment, you give it to the other person. Certainly, no one gave us information, and vice versa. You don't give information the day you do an experiment for multiple reasons. Yes, one has to do with some aspect of credit to your own colleagues, but also you have to be sure of your data, and you have to have, you want the data to reach the whole scientific community, i.e., you want it to be published. So all those things are part of the whole scientific culture. So, I mean, on the one hand, we have people saying it's the fastest progress in history. Lewis Thomas, the late Lewis Thomas, just wrote a book, The Fragile Species. You'll read that statement. Never in history has science moved so rapidly in that period of 83 to 85. Could it have been faster? I'm sure. Could anything be better? I'm sure. Of course there wasn't perfection on either side. But this is an absurdity to say that it could have gone even faster, maybe a week faster. I don't know how you could go any faster. One has to be specific of where it could have gone faster. One could say, you could argue that if in 1983, when we had some work done with the Pasteur virus, we had told them instantly what was done, they could have moved it faster. They couldn't possibly have we had the technology far more advanced than they did at that time. And we moved it fast. And I went to visit them before the paper was published in April 1984. So 1 month before it was published, they had the bulk of the information in hand, that we could grow the virus, that we knew how to do it, that we had many isolates, that the cause was known, that we had a blood test, was all told to them in the first week of April of 1984. You know when we got that data? We got that data on and off throughout 1983, but not until the end of 83 and the beginning of 1984 did we have evidence in our hands of causation and the blood test adequately developed. That means that our supposed major competitors had this information within 2 months of us. And the paper's published in May, all of them, 5 of them, 4 in Science and 1 in Lancet. It's pretty hard to have it more open and faster than that. So I think this is an absurdity.
I don't know, people have a naive impression that scientists should do things and not be driven by ambition, not be driven by the same that anybody else is. And ambition forces people to do things faster.
I have heard, but I've never heard him say it, but I've heard that somebody quoted Jim Watson saying, like, competition is not important, it's everything.
Right.
I don't believe he means it literally. It's not everything, of course. But it is important. And sometimes people want a cooperation, even when it's a nicety and it's false. Competition spurs people to do more, as long as it's reasonable.
Okay, so back in 1984, we have a blood test.
Right.
We now know that AIDS is caused by a virus,
Yes.
HIV. So now what is the general sentiment in the medical community in terms of when we might have a vaccine, when we might have a drug? I mean, there must have been optimism at this
Yes,
point.
there was. In fact, the optimism is opposite of the way things turned out. Namely, I don't think people thought there would be a drug right away because, remember, the history of virology is not exciting in terms of drug therapy for viruses.
Right.
There's
A
almost,
common cold.
there's almost no precedent. There's a little. I mean, we have some anti-herpes virus things that were about out at that time. I can't even remember when they came out. But antiviral therapy doesn't have a prominent history.
Let me ask you a very naive question. I mean,
Yes.
Why? Why can't we develop drugs for viruses
Because
like
the
the
virus is, as we said before, just a piece of genes with a protective coat that cannot live outside a cell, doesn't have its own metabolism, like a bacterium or a parasite, a protozoan parasite, or a fungus. It has no metabolism to target. It uses the cell's own machinery to replicate itself.
So in other words, to fight a bacteria, we can sort of break into its metabolism,
Sure.
stop it from eating.
And many specific targets in a bacterium. Whereas a virus, you have to be far more subtle, far more sophisticated, far more specific, and it's a whole new era of research in my mind. I think antiviral therapy will be mostly not what it is today with chemicals, but more from biological control. That we will learn how to harness some aspects of normal biology to harness viral infections that are persistent. AIDS is a chronic persisting virus. It's not unique in that respect. Those kind of viruses are going to require special approaches. And we'll come back to what we can do about that later if you wish. But that's the reason they're very hard to get at. And there hasn't been much of a field developed to target viruses.
Okay, so how about in terms of vaccine? Back in 1984, people
Yeah,
must
to go
have...
back to your point, I don't think that there was any Pollyanna attitude about therapy. But for preventive vaccine, I certainly thought we would make not easy, but rapid progress. Secretary Heckler at that time gave a few years as an estimate and was often been criticized for that. The blame should be, I think, mine. I didn't know anything about vaccinology. I'd never developed a vaccine. But it seemed to me when she asked me, well, here we could mass produce the virus. We have it in our hands. Shouldn't I start thinking it would take a few years, maybe, but we should have a vaccine in a few years. So I actually said that to her. Maybe I must not have been alone because she was seeking advice from a few quarters. When I think back about it, how naive I was, even if everything were easy, it would have taken time to prove it worked. But we simply meant that within a few years, vaccines would be available to try and probably begin to see efficacy. Okay, in fact, vaccines were tried within a few years. So you can say we were right, she was right, but obviously not with effect.
In a conventional vaccine, you give someone killed virus, right? And it stimulates their immune system so that if they're challenged by the real virus at some point, they're in red alert. You can't do this with AIDS, or you could do it, but isn't the danger that you might not kill all the virus and someone might get it from the vaccine?
Actually, it doesn't work anyway. In animal systems, when you use killed virus, it's been very ineffective, so we have no evidence it would work. I have taken the opinion that we can't use a live virus, even crippled, because we can't predict what would happen over a long period of time with a retrovirus, excuse me, integrating its genes into our DNA. There have been, I've already told you, the dead virus didn't work in animal systems. There's no evidence it gives a very good immune response for one reason or another. It doesn't seem to be a good vaccine approach. There are people who have argued for an attenuated live HIV vaccine. I think it's too dangerous.
Attenuated live, so a weakened live vaccine. Okay.
Right. Similar to Sabin polio
Right.
vaccine. However, they still integrate. And I believe it's too much of a potential danger to allow that to go forward. But that is a legitimate scientific debate. The argument some would make is that our backs are against the wall in some third world countries, and we better take a chance. Since the live, crippled, attenuated HIV does give a very good immune response. Its analog in monkeys, SIV, the simian AIDS virus, when crippled but kept live, can induce a very potent working vaccine. So the temptation is there. But I believe the risk is far too great in my mind. So I'll continue to argue against that.
And what stood in the way? I mean, it seems we know more about the AIDS virus than any other virus.
I think that is true. I think we know now more about HIV than we know about any other virus. But there are multiple things that stand in the way. I tried to summarize. First, it's a retrovirus. Do we have examples or precedent for success in an animal retrovirus? We have a little, not much. And difficulties are this. The retrovirus, as we discussed earlier, integrates its DNA into our chromosomes. So here, at least on paper, in theory, you want to block all virus infection. What happens with polio? As I understand it, the vaccine against polio does not block infection. You get infected. The virus replicates in the gastrointestinal tract. You stimulate your memory because you've been vaccinated. You get an immune response. You attack the virus. It's finished.
So it never gets out of your gut. Okay.
It never gets out of your gut to your brain and spinal cord. No polio. Let's say what happens with HIV. If we don't block all the virus particles from infecting, some slip through. They integrate. You knock them off with a good immune response, the rest. But there you have silent in your DNA the genes of the virus. And when the immune system is not highly active and the virus starts to become expressed, the virus returns. So you'd have to keep the immune system high all the time, or you have to have a vaccine that blocks 100% of infection. That's, on paper, what would be needed in theory to have a perfect vaccine. Second problem is any replication of this virus begins to damage the immune system, the very system that is attacking virus. Third problem is the variation of the virus, much more than other virus challenges, you know? So that
Changes very rapidly. Yeah.
changes, you know, you have so many subtypes, let us say, that will a vaccine be able to cover most? It is possible that we already have a vaccine against AIDS in our hand. We don't know it. So you'd say, why not go test it? That came up for discussion using what we call a vector-driven approach. This means that we have some genes of HIV, just a few, let us say, the gene for the envelope, the code of the virus, and you put it in another virus that's safe, a different virus, not an AIDS virus. You put the gene for HIV in this carrier virus. The carrier virus is a crippled one, but it gets into the cell properly and presents the viral coat protein envelope in the right configuration to be recognized by the immune system. And it gives an interesting immune response.
So you're just taking some harmless piece of
That's
the
right.
AIDS virus,
Right.
inserting that into some
carrier.
some carrier that's harmless.
And the carrier brings it in the right way, presents it in the right way, you stimulate the immune system,
Revs up your
and
immune
indeed
system.
sometimes you get protection in the monkey this way. So would that be a good thing to go forward with in humans? Well, let's go back to the animal data. Inconsistent results. No parameter we can predict that gave the protection. Strange. Usually not broad enough to be perfect. That is, it's not hitting enough of the variance. We'll
So what do we need to do? I mean,
I believe that if there's not some dramatic advance in the next few years, that increasingly major companies are going to walk away from this because there's poor incentives for vaccine. And I believe it's going to take true global cooperation, maybe even a taxation for this purpose, something very special. I believe we're going to have to put scientists with very good funding together in this problem with no pressures to publish, no pressures for their money.
poor incentives for companies to do this because it's going to take too long and too much money, and there
Very
are
costly.
too few people
Yeah,
who would benefit in the
dangers.
eyes of these companies. Yeah.
You have problems of danger, therefore insurance. You have problems of cost for production, failed experiments, no knowledge, not easy parameters to measure that they're going to have success. Not too many incentives to go forward. So I think this is one where we really need cooperation, not competition. This is where we really need people willing to talk about yesterday's experiment. I think not people that are needing support as young people, but established people who don't have to worry about publishing. I think we really need to bring together in one collective unity, maybe in a few buildings only, maybe in 3 or 4 or 5 buildings, linked and supported by some pharmaceutical companies working together, supported by the world. Maybe I don't know enough about economics and politics, but some tax for the whole world to do this. No pressure on the scientists, just come back at the end of a year and make a report.
If we could roll the clock ahead 10 years into the future, what kind of world are we going to face in terms of emerging viruses and new diseases?
I don't know. Is the honest and straight answer. Nothing would be surprising. That is, maybe nothing new and important.
I mean, something like the Ebola virus, which obviously grabbed headlines, it fizzled out very quickly.
Oh, yeah. I mean, you could have predicted that, though.
Right.
But if you had an Ebola that was easier to go around, of course, it would be a nightmare. If you had an AIDS easier to go around, it would be a nightmare. Could it happen? Theoretically, yes. Do I think it's going to happen? No, I don't. But that doesn't mean I'm going to be shocked if I get a surprise of still another virus. I don't think there'll be anything worse than AIDS. I don't believe there will be. But I can't say there's not going to be.
Right.
The best preparation we can have for it is to be prepared, is to be thinking of it. D.H. Henderson, great epidemiologist, the public health guy at Johns Hopkins, who played such a major role in the eradication of smallpox, has argued that we need surveillance centers at the edges of rainforests because of the changes and the possibility of not being able to predict what's coming. Ebola itself could be a nightmare even as it is.
I mean, aside from, the possibility of emerging diseases, which will either emerge or not, what are some of the big challenges that medical research faces in the next decade?
Chronic human diseases, of course, the overpopulation of the world and the overaging of part of the world, and all the chronic diseases associated with a large population growing of older people. You mentioned one, the possibility of another outbreak of an unpredictable nature. AIDS. The final attacks on cancer. The wealth of genetic information on human beings will have with the Genome Project and what to do with that wealth of information and using it only for good, which I'm sure will be the case. But it, as you know, presents periodically ethical dilemmas that have to be carefully sorted out and thought through. You asked me problems rather than breakthroughs in resolution.
Breakthroughs too, if you think
but I think the chronic diseases and the potential of the disasters from overpopulation that I can't predict easily. I mean, I don't, it's hard to foresee. But with big overpopulations in some areas of the world, we may create multiplicity of epidemics of kinds we can't exactly say right now. And we may have problems for food. And we may have economic disorder, which could create old bugs coming back. But it's hard to prophesize about those things. Our greatest challenges are going to come from the wealth of genetic information we have and in the handling of chronic diseases, in my mind, at least in the Western world.
Thank you very much, Dr. Gallo.
Thank you for having me.